All right, so to continue on with our program today, we have Yansheng Liu from Yale University, and I'll let him take it over from here.
Okay, good afternoon. So I would like to first thank, sorry, maybe I turn this off. So I would like to first thank Biognosis for this opportunity so that I can share with you some of our recent studies regarding quantifying and understanding phosphoproteome turnover from in vitro to in vivo. So as proteomic scientists, we normally measure a lot about protein abundance. However, we know that the protein abundance is actually the output of protein synthesis and protein degradation. And in this review, you can see that when we compare two mouse models, we normally can really ascribe the difference that we see into an abundance change, but often there is a turnover change along with it. So it would be very nice for us to figure out this abundance change and ascribe it to a change or regulation at the synthesis or degradation level. So I would like to further use this second slide to argue the importance about the interplay between phosphorylation as a modification and the protein turnover. So on the left, you can see this Wnt pathway, which is a classic cancer-related pathway. So in the absence of the Wnt signal, you can see this beta-catenin, which is the important Wnt protein, is get phosphorylated because of the activation of this APC complex. And this phosphorylation will render beta-catenin to be ubiquitinated and then degraded by the 26S proteasome. And when Wnt is binding to the physical receptor, this APC complex is inactive, so the beta-catenin cannot be phosphorylated anymore, and the unphosphorylated beta-catenin will be translocated into the nucleus to start the transcription of the genes that are promoting cell cycle and progression. So these are very important cancer-associated pathways, suggesting that we should understand better about how phosphorylation could regulate protein degradation for important cancer-related proteins. In the meantime, as the previous speaker also showed, that this phosphorylation for some neurodegenerative proteins could really cause the accumulation of these proteins to form these tangles or condensates that could also impact the effective protein clearance and protein degradation. So the question is that can we really degrade these hyperphosphorylated proteins, for example, for protein tau? So basically, if we understand better about the relationship between phosphorylation and protein turnover, we could maybe have better fundamental as well as translational implications. So as we know, the community is actually very powerful in measuring post-translational modifications using bottom-up proteomic approach. So we could use mass spectrometry to really identify these site-specific PTMs and their abundance and compare that between different conditions. So if you think about it, this is actually quite similar to the Western blot that people were doing for a long time. So basically, we can compare and understand the functions and abundance regulation between different biological conditions for this site-specific phosphorylation, site-specific PTM modification as well. So about protein turnover, actually the community also have a very powerful approach, which is called a DeltaSILAC DIA, a DeltaSILAC workflow. So in DeltaSILAC, basically we have the cells which are first cultured in the light medium, and upon a certain time point, we could then switch the light labeled medium with a heavy isotopic labeled medium, normally through, for example, heavy isotopic arginine and lysine. And then we can trace the incorporation of this heavy peptide into the human proteome. So basically, the newly synthesized proteins will be labeled with a heavy channel, while the pre-existing proteins are still in the light channel. So we could then use mass spectrometer to quantify these heavy to light ratios along the labeling time course and then use mathematical models to really quantify the protein-specific turnover rate or degradation rate. So we know that the DIA is very reproducible, and the DeltaSILAC experiment happened to be an experiment with multiple, that you need multiple samples due to the need of time course. So we reasoned that we could actually combine these two to really boost the identification and quantification for the proteins with turnover rate in the proteome scale. So I actually studied this combination when I was doing a postdoc in Rudy Episode group a few years already 10 years ago. And then after I joined Yale, in my independent lab, we actually greatly enhanced this research line. We looked into Lummer's Orbitrap setup to generate some method to optimize the instrument method. And we also generated some biological bioinformatic toolkits to facilitate the data analysis. And recently, we also applied it into different biological systems to understand some biological questions. So last year, for example, we have 3 papers published using this technique. So if you think about it, this is also very similar to Plex DIA from Nikolai's lab or MDIA from Matthias' lab. And this is really like you can really look at different channels of this DIA data to do some biological research. So since the proteomic approach are so powerful in measuring both post-translational modification as well as protein turnover, why don't we combine the two? So this is actually basically what we did. So in this paper we published a few years ago, we actually performed a quite simple experiment where we look at the proteome turnover for these HeLa cells. So we culture these HeLa cells in the light medium for a few days, and then we can switch this light medium into the heavy isotopic labeled medium, the SILAC heavy medium. So with these 5 labeling time point, we can perform the direct measurement of protein turnover using 5% of the peptide digest, as I was just told you. And also, we can then use the majority of the peptide to perform the phosphopeptide enrichment using phosphoproteomics. And the idea is that we can then look at the heavy to light ratio change of the phosphorylated version and the non-phosphorylated version of the same protein to infer how this heavy to light ratio change upon the phosphorylation. So what we find is that a peptide matching strategy could be really used to pinpoint this PTM site-specific turnover effect. So we find that if we can compare the same peptide regarding their naked peptide sequence to look at whether this peptide is phosphorylated or not, and really only compare the heavy to light ratios between these types of pairs, we could really pinpoint the turnover rate quite clearly. And this significant will be gone if you do not have this strictly match the peptide sequence. So we call this strategy DeltaSILAC. So, so once we apply this into these HeLa samples, we actually find that we can identify the phosphosite that can really render the protein to be degraded in a faster manner. But also we find other phosphosites that could really protect the protein from being degraded. So here you can see some mutagenesis analysis with Western blot to verify some of these sites. So this thing can be quite functional. So basically here we actually compared the lifetime difference between the phosphorylated and non-phosphorylated version of these proteins, and then divided these peptide or proteins into 5 segments based on the difference of the half-life time. And in Q1, you can see those phosphosites that are annotated to be in cell cycle or translation initiation. So for these phosphosites, they really tend to make the protein degradation to be faster. But on the other end, we actually find some other phosphosites which are often related to protein-protein interaction or the nuclear core protein complex or protein transportation. So this phosphosite, once happened, seems to prolong the protein lifetime or delay the protein turnover. So this can be quite interesting for us to think about the phosphoproteomic functions. So recently, we actually worked with Biognosys to establish further the bioinformatic toolkit, which is demonstrated in this paper. So you can see some names of our friends from Biognosys in the author list as well. So basically, we try to establish this robust multiplex DRA workflow from different aspects. So we first tried with different machines, like this Lumos Orbitrap we have, and also this Ultra mass spec from Booker. And we also tried to increase the identification rate by looking better into the heavy to light peaks that I will show you in the next slide. We also generated this flexible FDR controlling consideration so that we have flexible criteria to report the quantitative result. And we applied this in some standard samples with this 2-channel or 3-channel SILAC, and also in this cisplatin resistant cell model. So basically, if you look at this ultra data set, this is was quite new, but now is not the highest mass spec now, but you can see that we can quantify more than 9,100 proteins with their turnover rate reproducibly in a lot of samples. So regarding the identification in this type of approach, so basically, if you think about it, you could either rely on the heavy channel or the light channel, or both of them to do the identification. So previously, in also collaboration with Biognosys, we actually published in MSB in 2020 this ISW workflow to do the score of these SILAC pairs. So basically, the idea is that we will rely on the light channel to do the identification, and we rely on the heavy and the light channel to do the quantification. So this was found to actually increase the proteome coverage for protein turnover measurement. But if you think about it, with time goes by, you will have more heavy channel in your sample, you will have more heavy peptide in your sample, and that scoring might actually affect the identification for the later time point. So with the collaboration with Biognosys, we actually embraced the machine learning approach to better consider this. So basically, what we did is that we actually performed a dynamic identification. So the idea is that the machine learning will try to use more of the light features for those samples where you have more light peptide, and to use more of the heavy features to do the identification for those samples where you have more heavy peptide. So basically here you can see a standard dilution experiment by the heavy and light cell lysate mixed in different ratios. So you can see that with the traditional ISW workflow, indeed, we actually have a lower identification rate for those samples where we have a lot of heavy peptide. However, for the LBL workflow, you can see we consistently identified a lot of peptides that are comparable across this dilution curve. And with a spectronaunt, this relative scoring weight can be actually virtualized, where you can see indeed for those samples where you have a lot of heavy peptide, the weight of the heavy identification is much more. So by working with spectronaunt, we actually made this accessible since spectronaunt 19. This is actually already accessible, where you can really virtualize the weight of the scoring in different time points across this pulse-like experiment. And also for a particular protein, you can look at the heavy to light ratio of different peptide precursors for that protein. And then you can really see that with labeling, you see the increase of the heavy to light ratios along the labeling time. So these are very nice features that are supported in spectronaunt. So with this paper, we also put in some R package in which we compiled our in-house script to quantify the protein turnover rate. We call it a key dagger, and it is available in GitHub. So basically, this is this can be used to get the data input from spectronaunt and other softwares, and then we can perform the inference of protein turnover calculation, and then perform some data virtualization and comparison between different conditions, for example, in these two cell lines. So I hope this can be used in the future in the field to facilitate the data analysis in this regard. So I would also talk a little bit more about a recent study today, which we actually look at the proteome turnover and the phosphoproteome turnover in a lot of mouse tissues. So the idea here is that we can really use the diversity of the tissues as an important biological variable to understand better how these protein turnover rates are determined in the organism. So basically, in previous studies, there are certain papers already published to look at the protein turnover rate in multiple organs or tissues, but not in a lot of organs. So we hope to understand better by looking into more organs. And also, we want to look into this brain because there are a lot of different brain regions in this important organ that we could really dissect the variability. And we aim to use both DeltaSILAC DIA and DeltaSILAC TMT to boost the proteome coverage, and then to understand better about the determinants of protein turnover. So this study was a collaboration with Junming Peng's lab in St. Jude Hospital. Junming's lab actually provided this labeled SILAC mouse with different tissues, and then we applied our DIA approach, and also Junming's lab did this TMT analysis so that we can really quantify these protein turnover rates in a lot of tissues, actually 16 different mouse tissues. 9 of them are from the brain regions. So here we used a boxcar DIA. So this was following what my lab published about the boxcar method, but we are really doing it with the DIA and multiple injections with the purpose that we can actually have small windowed DIA so that we can split the heavy and the light peptide into different MS2 scans. And that by doing that, we could have a better quantification, and we can use all of the B ions for the SILAC quantification. And for TMT, we used a very extensive fractionation with 80 fractions and used a narrow M/Z isolation window-wise to really increase the quantitative accuracy with TMT. So because this is a mouse data, so we have to consider the free lysine turnover. So we used this JumpT software, which was published to really get this half-life time in hours and then quantify them for both DIA and TMT data set. So here you can see a data overview. Basically, with DIA and TMT, they agree a lot with each other if you look at the average lifetime quantified by both methods with R of 0.98. And we can quantify about 8,000 proteins in these brain regions with their turnover. And for the identification, we actually can do even better. So we then qualified our data with some previously published data. So in this data, you can see from the Venn diagram, we can see that our data set actually reported much more protein numbers with turnover rate quantified. And if you look at this half-life time, the correlation is quite good for these overlapping proteins between the two studies. And here you can see the DIA intensity. So basically, if you look at the identified proteins, you can see that actually we can actually identify more simply because we can go lower with the proteome depths. So with this, we can actually make this data set available. So we actually used this Shining app in my lab and generated this tissue PPT to look at this turnover rate of different proteins across different mouse tissues. We call it a turnover atlas. So for a long time, I actually want to virtualize both protein abundance and the protein lifetime. And we actually managed to do this using this heater scatter plot, which I think is quite nice. So basically here, each node denotes a protein, and we can then use the size of the node to denote the abundance of the protein, while the color of the node from the red to the blue to denote the short-lived protein or the long-lived protein. So then we can do this type of virtualization for different proteins, for different organelles, or for different tissues, so that we can really navigate and understand better how the protein turnover and abundance are and their relationship across these tissues. For example, for this collagen protein, you can see that their abundance variability is very high between tissues. However, we see that they are all long-lived, suggesting that they may have this structure supporting role. And for these SLC proteins, we know that they are very important membrane proteins. We can see certain proteins are really outliers with their longer lifetime across tissues. So this will be very helpful for us to understand protein-specific functions. So here you can see we zoom in with some important protein set with this HC plot. So basically, these are the protein removal process. We can see that proteins related to the proteasome are quite tightly regulated for both abundance and turnover across these mouse tissues. And for the lysosome, you can see that their abundance range is much variable, and also the lifetime is also quite more different compared to the proteasome level. And interestingly, if you look at this E3 ligase and their accessory proteins, you can see that they are in general low abundant, and also the lifetime is quite diverse between different mouse tissues. This suggests that if you consider the tissue diversity, the E3 ligase lifetime can be very important. So with this data, we could also map the protein lifetime with protein-protein interaction, which is also an interesting consideration to look at the protein function. So we actually mapped the lifetime data with the Quorum database, and here we performed the protein pair-wise correlation analysis, and we find that those subunit of the same Quorum complex are actually very well correlated with their lifetime across different tissues. And this is of course expected, but we actually get even better result once we map this correlation to the tissue PPI dataset. So this dataset is actually from Leonard Kuster's group that they actually used the size exclusion chromatography to fractionate these proteins from different mouse tissues, and then use PCP or a protein correlation profiling to infer the protein-protein interaction. So this is better resolved because we can then compare the same tissue with their PPI. So here you can see that we were a little bit surprised to find that the lifetime correlation between these protein pairs is even better than the correlation between the abundance of these protein pairs. So this is somehow not expected, because this actually indicates that the lifetime is a very strong indication of the protein-protein interaction. So we even performed some ROC curve analysis to ask the question that whether or not we could rely on the lifetime to predict the protein-protein interaction. And you can see the AUC is 0.82, suggests that this is indeed a very strong indicator for protein-protein interaction. And if we combine the abundance and the lifetime measurement, we could have the AUC of 0.89. So this is very interesting for us to keep in mind when we understand protein-protein interaction. So with this data, we could also map this lifetime diversity into different organelles. So here, because this is mouse tissue, there are a lot of data about mouse tissue. So we also compare the data set where they measured the translatome using the ribosome profiling to look at the translation rate. So then we can perform this multi-omics correlation analysis for different organelles. So interestingly, you can see that this MI protein half-life correlation is quite weak in a lot of organelles, with the exception of peroxisome. So here you can see the mRNA abundance, protein abundance for the peroxisome proteins distributed in these 5 tissues. And if you look at the lifetime, you can see that this is indeed in a reverse trend compared to the protein abundance. So this, of course, can be driven by some important enzymes in the liver and their short lifetime. But in general, I think it very interestingly suggests that there is a strong turnover control for the peroxisome proteins that was not reported to the best of our knowledge. So again, we can then go back to the phosphoproteomic analysis. So since it's very easy for us to do a phosphoproteomics using the same data set. So here you can see we identified 500 proteins that 500 phosphosites that tend to stabilize the protein, and a similar number of sites that tend to destabilize the protein in these brain regions, if you look at all of the brain regions. And some of them are really related to the phosphorylation of important neurodegenerative proteins, although this is a health mouse model. So we then did this verification. So one verification we did is based on this PhosTag, which is actually a molecular glue developed by Craig Ku's lab, which is also at Yale. And we used this PhosTag to treat the in vitro cell cultures. The idea of the PhosTag is that it will try to recruit the phosphatase to really perform the targeted dephosphorylation. So here we use the PhosTag for tau, and that PhosTag actually removed the tau phosphorylation, and in the meantime, we can observe that the heavy to light ratio is indeed higher upon the removal of phosphorylation. So this suggests that there might be some translational implication here. And we also used this high content imaging to and mutagenesis for the alpha-synuclein and the tau phosphosite. And we verified that a certain phosphorylation site of these proteins could indeed impact protein turnover, actually prolong the protein lifetime. So this brings me to the summary. So I hope I showed you today that the DeltaSILAC DIA actually effectively measures the in vitro and the in vivo protein turnover and become more efficient if we combine the latest bioinformatic advances and also, of course, the mass spec instrument advances. And we generated this turnover PPT, which is a comprehensive resource for us to understand the lifetime and abundance regulation for more than 11,000 proteins and 40,000 phosphosites in mouse tissues. And I think it's also interesting for a phosphoproteomic perspective, because we know about phosphorylation. Normally, we care a lot about kinase and phosphatase, this short-term regulation of phosphorylation. But in the longer term, this approach might be able to help us to find certain phosphosites that could impact the protein stability and expression, and that could be some new research line to think about in the future. So with this, I want to thank the lab, especially Wen Xue, who lead the tissue PPT study, and all the lab members, especially our collaborator, Junbin Peng's lab in St. Jude, for the collaboration, and Eugenol in Göttingen for a constant discussion about protein turnover, and of course the diagnosis team and my funding. And thank you for your attention. I'm here for questions if you have any.
Thanks for the great talk. Here.
Oh yeah.
Yeah. 2 questions. First, could you clarify, to switch in between ISW to LBL? Is it pure computationally, or in the wet lab, we also need to take care of some parts? That's the first one. And then the second one is, if it's possible for you to comment or talk a little bit about how the machine learning help the heavy light on the peptide application. Thanks.
Yeah, so I don't know if I can go back quickly. So to answer your question, in short, actually this is purely computational. So for the same data set, you could choose to only use the light to do the identification, or use both heavy and light channel to do the identification. But previously, because we focus a lot on DeltaSILAC, right? So we care a lot about those samples with more light signal. So that's why we have this ISW workflow. Recently, with the labeled workflow, we can see this is actually not only working for the DeltaSILAC sample, but also for the SILAC sample, it will also work. Or for the MDIA sample or the plex DIA sample, because in that cases, you actually do not need to assume that light is always higher than heavy. So basically, this is all computational. And how does that work is really related to the machine learning approach that if you think about it, if you flip, for example, heavy into the light, basically you have a lot of transitions from the heavy channel, from the light channel. Both of them can be used to identify the peptide. So in that case, the identification rate is actually very high. But however, we also have the FDR to look at the quantification. So in that case, you can focus on the mini Q or the max Q, where you really look at the channel-specific identification, or you really require both channels to be quantified. So this is actually quite flexible, and in general, it works quite well.
So the machine learning means like compared to the traditional, like identify in either channel, do you like require it to be combined and then
Yeah, previously we only look at the light channel for identification. Now you can do both. Yeah.
Okay. Thanks.
Sure.
Hi, I have a question over here.
Sure.
Great talk. Can you please elaborate a little bit more about the phosphostack technology that you use, and if this phosphostack indeed impacted the degradation of tau synuclein, and then to what degree?
Yes, so phosphostack was a molecular glue type of molecular glue developed by Chris Ku's lab, and we actually had some collaboration that we participated in two of their papers where they try to establish for protein tau and for EGFR. However, these are still under development. So the idea is that you can then recruit phosphatase. For example, for EGFR, this can be tyrosine phosphatase. And then you can really remove the targeted phosphorylation. And here you can see this is actually in vitro validation in cell line models. So basically, once you remove the phosphorylation, indeed you see that the heavy to light ratio is regulated a bit upon the molecular glue. Yeah. But this is quite preliminary, I would say. Yeah.
Okay, so thank you very much, Yansheng, for your talk.